Okay, hi guys, so today we are going to do a recap on excretion in humans. Okay, so this video will focus on the processes in which urine is being produced, okay, in aid of this process called excretion. Okay, so when we talk about excretion, we always have to look at this particular organ. Okay, so this is actually a part of the kidney. This structure is actually the nephron. Okay, so to orientate you a bit, the one in red color, the tubes in red color are your blood vessels. Okay, and then the orange color ones are parts of your nephron. Okay, so now let's do a quick recap of the different parts of the nephron first. So this is the bowl-like structure or cap-like structure, okay, and we call this the Bowman's capsule. So in this particular website, it's known as the renal capsule, but it's okay, we'll call it the Bowman's capsule. So Bowman's capsule, as it goes down further, this first part where it's very windy and long, okay, it's called the proximal convoluted tubule. So proximal means that it's in close proximity to your Bowman's capsule. Okay, so as you go down further, you see this U-shaped tube, and this is actually your loop of Henle. Okay, and then as you go up further, you encounter another region of convolution. This is your distal convoluted tubule. Distal meaning that it's further away from your Bowman's capsule. And then finally leading into the collecting duct. Okay, so this is actually your collecting duct. So as you notice, the collecting duct has many branches, meaning that many nephrons actually connect to one collecting duct. So they actually share a collecting duct. Okay. So orientate you a bit with regards to the blood vessels. So at this portion here, blood vessel going in, this entire network of blood vessels in your Bowman's capsule, this is called the glomerulus. Together, the Bowman's capsule together with your glomerulus, this is called the Malpighian corpuscle. Okay, so if you notice, the blood vessel going in is much thicker than the blood vessel coming out. So the blood vessel going in is called your afferent arteriole, starting with A, A-F-F-E-R-E-N-T, afferent, and the blood vessel coming out will be your efferent arteriole, E-F-F-E-R-E-N-T, efferent arteriole. Okay, so with this brief recap of your structure, we'll now move into how urine is actually being formed. Okay, so formation of urine in your kidney is actually down to two main processes that occur. Okay, so two main urine production processes are your ultrafiltration as well as your selective reabsorption. So the first process that occurs would be your ultrafiltration, and as the name implies, filtration. Okay, so it involves the removal of small molecules from the blood, and these small molecules will be able to pass from your blood vessels in the glomerulus into your Bowman's capsule in your nephron. So let's see what exactly happens. Okay, so in ultrafiltration, as mentioned just now, it happens in your Bowman's capsule, which is this portion here. So now we're going to zoom in, okay, into this portion to see what exactly happens. Okay, so this is a zoom-in version of your Bowman's capsule. This is obviously your red blood cell, this is your blood, and this is your basement membrane separating your blood vessel, your glomerulus, and your your Bowman's capsule in your nephron. Okay, so this is your space inside your Bowman's capsule. So let's see what's happening here. Initially in your blood, you have many different kinds of molecules, right? Like your urea, your water, glucose, protein, sodium, and so on. Okay, but what actually happens here, if you notice carefully from this animation, only some molecules can pass through from your blood into your Bowman's capsule, whereas some will actually remain in your blood. So specifically, your protein, the green color molecule, and your blood cells are very, very big, and so they will not be able to pass through your basement membrane, the pores in your basement membrane. So they will be contained within your blood still. Only small molecules like your urea, like your glucose, like your sodium, like your water molecules are able to be filtered because they're very small. They're able to pass through the pores in your basement membrane into your space in your Bowman's capsule. Okay, so this process is called ultrafiltration. Okay, so in this case here, during ultrafiltration, most of the water, most of the minerals, most of the nutrients are being filtered into your nephron. Okay, so going back into your structure of your nephron, so most of your water and nutrients and minerals are filtered from your blood into your Bowman's capsule, into the nephron at this particular point. Okay, so to recap what we saw just now, ultrafiltration, it occurs in your Bowman's capsule or your renal capsule, okay? And the entire structure is called your Malpighian corpuscle. Okay, so why can ultrafiltration occur? It's mainly because of two different factors. Okay, firstly, is because your afferent arteriole, as seen in the video just now, is much wider. Okay, the lumen is much wider than your efferent arteriole. So as a result, this creates very high blood pressure at the glomerulus, which will force water and molecules out of the blood, okay, through the basement membrane, the porous basement membrane, into your Bowman's capsule. Okay, so if you just were to imagine your afferent arteriole is very, very thick. Okay, it's very, very thick. So it means that there are a lot of blood molecules, a lot of molecules flowing through here, okay, flowing into your glomerulus. But at the same time, the exit is very, very small. The lumen of the efferent arteriole is very, very small. So it means that the exit of your molecules in the blood, it happens at a much slower rate. So if you were to just think about it, when molecules enter at a faster rate as compared to the rate at which it's able to exit, this will cause a build-up of pressure inside this region here, because entry is much faster than your exit because of the difference in lumen size of the afferent and efferent arteriole. So this results in a build-up of hydrostatic pressure within your glomerulus here, okay, which is one of the driving force for ultrafiltration. Okay, so filtration basically means that there is a partially permeable membrane that surrounds your glomerular blood capillaries and acts as a very fine filter, so meaning that it's very, very porous. And porous is partially permeable, so only small molecules like your water and nutrients can pass through, whereas big molecules like your blood cells and your proteins will not be able to pass through and will remain in your blood vessel, blood capillary. Okay, so just to recap, which are the molecules that are able to pass through your blood vessel? Okay, so water, you know that it's a very big molecule, so it will be able, so it's a very small molecule, so it will remain, so it will be able to pass through into your, you'll be able to be filtered. Mineral salts, also small molecules, so it's able to be filtered into your nephron. Fats, they are very big molecules, so they will not be able to be filtered. Proteins are big as well. Red blood cells are big. Nitrogenous waste, they are small molecules. Amino acids, they are small. Glucose are small. And your white blood cells are big molecules. So only these molecules will be able to be filtered. Okay? Okay, so that is the first part, which is ultrafiltration. So after ultrafiltration here, most of our water is actually being filtered into the nephron. So is this ideal at all? Would you want to lose all of your water that you drink into as part of your urine? Okay, so this is not technically not possible. So because a lot of water is required for our life processes as well as to maintain our cell turgidity and so on. Okay, so of course during the later parts of our nephron, what will happen is that some of the water, the essential water, as well as nutrients, will be taken back. So this process whereby nutrients and water, which are filtered here, are reabsorbed, this is called selective reabsorption. So let's see what happens in your proximal convoluted tubule first. Okay, so this is a zoom in of your proximal convoluted tubule. Okay, this is your blood vessel, this is your proximal convoluted tubule, the lining of it. So if you notice, okay, so in your lining here, initially from the ultrafiltration stage, you have your urea, you have water, you have your glucose as well as sodium that has been filtered. So what happens here is that, if you notice, urea is not being filtered back. So only water, glucose and sodium, they are being reabsorbed back into your bloodstream. Okay, so essentially all the essential nutrients that you want, that you don't want to lose, you'll reabsorb them back. Okay, so in this case here, most of your water molecules are reabsorbed at this particular point in the proximal convoluted tubule. Okay, all of your glucose as well as amino acids will be reabsorbed at this point. Sodium also, most of it will be reabsorbed into your blood at this particular point. Okay, so the process in which they are being reabsorbed, this will give you a clue. So what are these molecules? These are actually your, what are these structures, sorry. So these are actually your mitochondria. Mitochondria for what? To release large amount of energy for active transport. Okay, so all these molecules, that mainly your glucose and your sodium, okay, they are being actively transported back, okay, into your bloodstream. For water, it is via the process of osmosis. Okay, so this is at the proximal convoluted tubule. So as we go down further into the loop of Henle, more molecules are being reabsorbed back. So let's see what happens in the loop of Henle. Okay, so in the loop of Henle, you actually have your water molecules mainly that is being reabsorbed back. So some of your water molecules are being reabsorbed back into the bloodstream at this particular loop. Okay, that's what mainly happens. And as you go down further into your distal convoluted tubule and you're collecting that, okay, we see that your water molecules, okay, are being able to reabsorb back, as well as some sodium molecules as well. Okay, so to consolidate what we have looked at so far, okay, we understand that there is actually a lot of filtrate being formed as a result of ultrafiltration, namely 120 cm3 of filtrate each minute. That is around 5 liters of blood. Okay, 5 liters being filtered. So that is definitely, you know for sure that you don't lose 5 liters of water through your urine every day. Okay, so definitely some of the useful substances such as water and nutrients will be reabsorbed back into the bloodstream after ultrafiltration. Okay, so these are the small molecules that are filtered into your nephron, into your Bowman's capsule. But of course, some of these materials are useful and we want them back. So how do we get them back? It's through the process of selective reabsorption, where useful materials like water, mineral salts, amino acids, glucose are being reabsorbed back, whereas your excess water, excess mineral salts, as well as your waste materials are not reabsorbed back. So in this case, because some are reabsorbed, some are not reabsorbed, it is a selective process. Okay, so this is a recap of what's reabsorbed where. So we understand that selective reabsorption mainly occurs after the ultrafiltration has occurred. That means after this particular portion. So it starts from your proximal convoluted tubule. So at your PCT, most mineral salts, most water, and all your glucose and amino acids are reabsorbed here. Okay. So in a healthy person, most of it, all of the glucose and amino acids are reabsorbed here. So in the case where a person has a faulty kidney, has some kidney disease, okay, one indicator would be that there is glucose found in the urine, okay, because this selective reabsorption process cannot occur properly. So this will lead to diseases like your diabetes and so on, which we will cover in a later part. Okay, so selective reabsorption occurs via osmosis for water and active transport for your mineral salts, glucose and amino acids. Okay, so for loop of Henle, the water leaves the loop of Henle to concentrate your urine. So over here, mainly water is being reabsorbed back at this particular point. Okay. And at your distal convoluted tubule, it is mainly your water as well as mineral salts. Okay, and finally, in your collecting duct, it will be your water, excess water. Okay, sorry, water. So all the excess water, mineral salts, as well as your nitrogenous waste will flow through and exit as urine. So this is a good summary slide that will tell you what is reabsorbed at which point. Okay, so over here, all glucose, all amino acids, large amount of water, large amount of mineral salts. Over here, loop of Henle, water. At your distal convoluted tubule, water and mineral salts. At your collecting duct, water. Okay, and what comes out as your urine will be your excess water, excess mineral salts, as well as your nitrogenous waste. Okay, that's all.